The release of hydrochloric acid from parietal cells of the stomach are as follows. In yellow here is a parietal cell. This right here is a hydrogen potassium ATPase. This is a chloride channel. This is a protein that functions as a cotransporter, specifically an antiporter. This region up here would be the lumen of the stomach, and we have a capillary down here. So the first thing that's going to happen is water is going to dissociate into the hydrogen ion and the hydroxide ion. The hydroxide is going to react with carbon dioxide, which is going to make bicarbonate. Bicarbonate is going to leave the parietal cell in exchange for chloride, which is the chloride shift exactly as we saw it in red blood cells for the transportation of CO2. This bicarbonate is going to move into the bloodstream and create what's known as an alkaline tide, a basic pH of the blood leaving. So once again, water dissociates into the hydrogen ion and hydroxide. Hydroxide reacts with CO2 to make bicarbonate. Bicarbonate leaves a parietal cell in exchange for chloride. That hydrogen is going to be pumped against its gradient via the hydrogen potassium ATPase. The lumen of the stomach can be as low as 0.8, a pH of 0.8, and the pH within the parietal cell is about 7.2. So it's much, much more acidic in the lumen, which means the hydrogen ion is moving against its gradients, moving into the lumen. It's actually moving against a very significant gradient, hence the reason we need energy in the form of ATP to pump hydrogen across the apical surface of the parietal cell into the lumen. This chloride, once it's in the cell, is going to proceed out the chloride channel into the lumen. So now we have chloride, we have that hydrogen ion out here, and that's how we get the release of hydrochloric acid from parietal cells into the lumen of the stomach.